I'll continue with our topic, that is biotechnology. And yesterday, when we started with biotechnology, we had first and foremost understood some of the basics related to biotechnology, and then we had moved on to various different branches of biotechnology. All right. So to continue from there, what we discussed was about agriculture, what we discussed was about medical applications, and we had discussed blue biotechnology and industrial applications. So these are the four application areas that we had discussed. And then after that, we come to a very crucial area of importance with regards to biotechnology, and that is the use of biotechnology in the environment. Something that we also call as gray biotechnology, as we had seen yesterday. Now, when we use biotechnology in the environment, this is largely for either the cleaning of the environment or looking at some potential benefits environmentally of biotechnology. So that is where we talk about a term called as bioremediation. Now, first and foremost, we'll try to understand what is bioremediation and what are the different ways in which we can make the use of bioremediation. And this is where biotechnology finds some use in environment. Now, when we say bioremediation, again, it is something, a term that we can try to break down and try to understand what exactly that term means. So, bioremediation. So, in a manner, biological remedy, it's some kind of remedy biologically is what we are trying to find. So that's why, in a manner, we are trying to solve environmental problems with the help of living organisms. All right. So that is basically what we call as bioremediation, and that's why it is cleaning of the environment with the help of living organisms. Cleaning of the environment with the help of living organisms will be called as bioremediation. Now, again, here there are two types of bioremediation. One is called as in situ bioremediation, and the second one is called as ex situ bioremediation. In situ bioremediation, the term in situ, the meaning of the term in situ is at the site. At the site. And the term ex situ means away from the site. So in situ means at the site and ex situ means away from the site. Now, in this manner, when we say in situ bioremediation techniques, we mean that this is the kind of bioremediation that will be carried on at the site of contamination. Meaning, wherever contamination has happened, wherever you see some kind of pollution, we are trying to solve the problem there itself. So, then in that case, it is called as in situ bioremediation. On the other hand, we have ex situ bioremediation, where whatever contamination has happened, we first take that contamination away from the site of contamination and we treat it there. All right. So that's why when you treat it at the site of contamination itself, you'll say that it is in situ bioremediation. And when you do the treatment away from the site of contamination, you will say that it is ex situ bioremediation. So first and foremost, when we talk about in situ bioremediation, what do we mean by in situ? Let's try to understand that. So as I told you that this is the treatment of the waste at the site of contamination itself. And here there are multiple different techniques that could be used for this kind of treatment. So try to understand what these techniques are. We have something called as biosparging, bioventing, bioaugmentation, bioculture. All these kind of techniques are used in case of bioremediation. Now, when it comes to all these techniques, we'll try to understand what these techniques are. So first, and there are two important techniques that we have to learn here. One is called as biosparging, and then the second one is called as bioventing. So what do we mean by biosparging and what do we mean by bioventing? We'll try to understand that. So first technique is biosparging. Now both these techniques, biosparging and bioventing, they are very similar techniques. All right, they're very similar techniques, and there are small differences between these two techniques that we have. So let's try to understand what the differences are and what is biosparging versus what is bioventing. So first and foremost, let's say if we talk about the layers of soil. So suppose that there is this top layer of the soil versus this is a layer of soil or layer of piece of land which is little below. And of course, below this, we will have the water table, correct? Below this, we'll have the water table. So you have water here. And we have top layer and the bottom layer of the soil. Now, when we are looking at these two layers of soil or layers of piece of land, this is where we need to understand the difference between the two. So whether it is the top layer or whether it is the bottom layer, will already have some of the microorganisms present, correct? Some of the decomposers will already be present at these sites, right? These decomposers, they will already be present at the site. So these are the decomposers or the microbes, right? So they are already present, correct? Whatever be the layer of the soil, they are already present, and a natural decomposition will keep on happening, right? Natural decomposition will keep on happening, but that natural rate might be pretty slow. Natural rate of decomposition might be very slow, so we want the rate of decomposition to be faster. Now, how do we ensure that the decomposition being done by the decomposers is faster in its rate? To make it faster, we provide them with some energy, with some food. So we basically feed them. You feed them, they gain energy, and they'll work faster. So in that manner, they'll be able to do the decontamination process much faster. So that's the reason. What we'll do is that we will insert some nutrition. We'll insert some nutrition. So nutrition will be inserted to this level, also at this level. So nutrition will be inserted. Now, this nutrition, so what we are inserting is nutrition plus oxygen. Nutrition plus oxygen. Oxygen will help them in better respiration, and that's why the process will be faster. Process of consumption of energy and the use of that energy will be faster. So that's why nutrition and oxygen will be injected to this layer. So as you can see, that the decontamination process that we are carrying out, the bioremediation process that we are carrying out, we are carrying out at the same place where the contamination had happened. We already know that microorganisms or the decomposers will be present, and there will be a natural decomposition that will keep happening, but we need this rate of decomposition to be faster, and for that we just provide them with extra nutrition and extra amount of oxygen, and with that we'll see that their process will become faster. Now when this is being done, so in both the cases, whether it is biosparging or whether it is bioventing, in both the cases, it is done in a very similar manner. Just the basic difference being this top layer of the soil, sorry, this biosparging is something that we do in the saturated zone, versus when it comes to bioventing, we do it in the unsaturated zone. So the difference is that when we say saturated zone or the unsaturated zone, this bottom layer, this is basically called as the saturated zone. This layer is called as the saturated zone. And this layer is called as the unsaturated zone. And for a very simple reason. Just think of a reason. Why do you think that this layer, the bottom layer, is being called as saturated, and why are we calling the top layer as the unsaturated zone? Just take a guess. Yes, Garvit, you have given the correct answer. Vashna correct. Nikki correct. Vivek correct. So you all have been giving a lot of you below that. So you have been given the correct answer. That because it is near water, that's why we are calling it as saturated zone, because it does not have the capacity to absorb more moisture, right? it will not be able to absorb more moisture, and hence it is being called as the saturated zone. Whereas the top layer of the soil is away from the source of water, and also there will be evaporation because of the sunlight. So that's why it has more capacity to absorb moisture, and hence it is being called as the unsaturated zone. So now when this process is being carried out in the unsaturated zone, we say that it is bioventing. And when this is being carried out in the saturated zone, we say that it is biosparging. That's the difference between biosparging and bioventing. So both, as you can see, that both of these methods are pretty similar methods. The very basic difference being that one is being carried out at the top layer and one is being carried out at the bottom layer. And because biosparging, so I'll just write it here. So the one that we are doing in the unsaturated zone is called as bioventing. This is called as bioventing. And one being carried out in the saturated zone is called as biosparging. All right, it is called as biosparging. So the what will be the difference between biosparging and bioventing? That because bioventing is happening in the unsaturated zone, it's easier to carry out that process. I mean, it's easier to inject the nutrients and oxygen to the top layer. Compared to that, when you're going down below, it's not going to be easy for you. And that's why this is something that we'll do at high pressure. So we'll have to exert more pressure so that it can go to that level. Whereas carrying this same process in the unsaturated zone via bioventing will be much easier, because it's there in the top layer of the soil. So it can be done in at low pressure as well. Whereas in case of biosparging, we'll have to do that at high pressure. So that's the basic difference between the top layer and the bottom layer of the soil. So as you can see, in biosparging, oxygen and nutrients are injected at high pressure to increase the biological activity of the indigenous microorganisms. Whereas when it comes to bioventing, in case of bioventing, we will be introducing the nutrients and oxygen at low pressure. So that's the basic difference, that the addition of the nutrients or air and oxygen will be done at low pressure in case of bioventing. So I hope this is clear. So that's why this is happening at the site of the contamination itself, just that at two different places in two different layers of the land. Many a times, I mean, these kind of methods can be used in agricultural lands, but largely we see that in industrial applications it is used more. Wherever land contamination might have happened because of some kind of industrial activities, we see that biosparging and venting methods can be used in those kind of cases. So that's why these two become very very crucial. Apart from that, we also had a mention of two other techniques. You have bio augmentation and bioculture. So bio augmentation is, as an example, is what we had discussed yesterday of oil zapper. So what we had discussed about oil zapper is that oil zapper is a formulation that consists of 5 different types of bacteria, and we take this formulation to the site of contamination on the sea surfaces or ocean surfaces wherever oil spills have happened. Correct. So that is basically what bioaugmentation is. So bioaugmentation is when you are taking the microorganisms to the site of contamination. In biosparging and bioventing, the microorganisms were already present, just that you wanted to enhance their activities. Correct. But in case of bioaugmentation and even bioculture, you are basically taking the microorganisms to the site of contamination, but yet the contamination is being treated at the site of contamination itself. All right. So that's why in case of bios augmentation and bioculture, we take the microorganisms to the site of contamination. So that is the basic difference. That here we'll see that this will be taken to the site of contamination, whereas in case of biosparging and bioventing, they are already present, just that you are trying to enhance the activities. So this is what happens in situ bioremediation. After this, we have ex situ bioremediation. So what ex situ bioremediation says is that we will be transferring the contaminated material for treatment to some other site. That is, you'll take the contamination or the pollution away from the site of contamination and do the treatment there. So this is what we do with ex situ bioremediation. So here, for example, you have land farming, where we'll prepare the bed of soil and periodically the tillage of the soil will be done. And with the tillage of the soil, we basically try to wake up the microorganisms. Again, the microorganisms are present already, just that they have slept and you just want to wake them up. And for that, tilling of the soil is done. So periodic tilling of the soil will help in waking these microorganisms up, and then after that they'll be treating the contamination on the soil. So that's why in this particular scenario we see that the soil is periodically tilled to stimulate the growth of microorganisms for degradation of the organic waste. So that is basically what happens in land farming. So easy technique, just that you take the soil to some part, some place, and there periodically tillage of the soil will happen, and that will just wake up the microorganisms so that they can start their decomposition process. Then after this, we have something called as biopile. Now, in case of biopile, the method is much more intricate. It takes a lot more time and complexity is higher, but the results are also better compared to just what happens in land farming. We take one step further from land farming, and we also use the composting techniques here. So that's why we say that it's a hybrid of land farming and land composting, where each and every property of the soil, each and every property of the soil is something that we try to control. For example, let's say any crop, it needs certain amount of moisture in it, it needs a certain pH value, it needs a certain type of soil, it needs a certain amount of nutrition in it. So all these requirements, whatever we have as requirements for a particular kind of farming, all these requirements will be met, and that's why it takes a lot more time, and it's a much more meticulous process. So that's why moisture, heat, nutrients, oxygen, pH, all these things are controlled so that we can enhance the biodegradation process in a manner that we can produce a much better quality of soil. And that's why the treatment of this kind of soil or this kind of a treatment will take a lot more time, and it can take up to 3 to 6 months typically. All right, it can take up to 3 to 6 months of time for this biopile to be ready, and once it is ready, then it can be shipped off. So that's why we see that this will be a much better quality of soil as compared to what happens in other cases. Then after that, there is also something called as phytoremediation. And yesterday also we had discussed about this term phyto. Phyto means plants. So whenever we are making the use of plants for any kind of bioremediation, we'll say that it is phytoremediation. Now, of course, phytoremediation as a process, uh, you'll see that, I mean, you can in a manner say that every plant takes in carbon dioxide and gives out oxygen. So you can say that phytoremediation is a natural part of a plant's being, correct? But there are plants, there are trees which go beyond this. For example, there are plants and trees which are known for their medicinal properties. They are known for their cleaning properties. For example, the neem. We know that neem plant is a very good example of phytoremediation, because it will be able to absorb the poisonous elements and reduces the growth of harmful microorganisms. So that's why naturally this is something that can be used for phytoremediation. Similarly, we see that there is a sub portion, a sub a part of phytoremediation called as phyto extraction. So when we say phyto extraction, meaning is that it is trying to extract something out of the soil. So many of the roots of the plants, what we do is that if let's say there is a kind of soil which is contaminated, there's a soil, some part of soil which is contaminated, so we'll plant the trees there, we'll plant some shrubs there, so that the roots of these trees and the shrubs, they will keep on uh, absorbing everything from the soil, absorbing all the poisonous metals and heavy metals, etc., from the soil, and they will be able to help in phytoremediation. So that's why this kind of a process where they are extracting all the contaminants away from the soil is called as phyto extraction. So that's why the plants will remove the dangerous elements or compounds from the soil, and mostly heavy metals or metals that have high density, we'll see that they will all be extracted away by largely the roots of the plants. Right, largely this will be done by the roots of the plants. So that's why we see that this is something that can become very, very crucial. Apart from that, there is another term that we use, and this is myco remediation. Now again, the term myco, when we are using it, basically involves the use of fungus, or what we call in its plural form as fungi or fungi or fungi or whatever you want to call. Just that you have to write the correct spelling. Nobody is going to ask for your pronunciation of it. So that's why a safer term to use is fungus. Nobody will question the term fungus. So whenever we are making the use of fungus for any kind of remediation technique, you will say that it is mycoremediation. And we know that they are very, very useful, especially in agriculture, because they also help in nitrogen fixation. Some of them can also help in nitrogen fixation. Now nitrogen fixation, they will help in, basically they will be able to enhance the quality of soil. They will be able to enhance the quality of soil, basically enhance the quality of soil, basically they will by adding nitrates. By adding nitrates to the soil. So in this manner, they will be able to help in nitrogen fixation, and this kind of activity will be called as myco remediation. So in these kind of cases, especially when it comes to phytoremediation and phytoextraction, etc., we see that in removing heavy metals or metals that have high density, they are useful largely because microorganisms are very difficult to use here. Because microorganisms will not be easily able to break these heavy metals down. So that's why the better method is just to extract. Because in other cases, what we see is that microorganisms go there and they start their decomposition activity. But here we are not talking about that decomposition activity, because it's very difficult for them to break these heavy metals down, and hence we'll simply try to extract these heavy metals away with the help of the roots of the plants. So that's why all these methods, they become very crucial in the cleaning of the environment. Read this question. C and D options. So let me clarify. In the context of solving pollution problems, what is or are the advantage or advantages of bioremediation technique? First, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. This is precisely what we discussed in biosparging and bioventing, that naturally also the biodegradation process would have happened and the decontamination would have been done by the microorganisms, just that a natural process would have been very slow, and we don't have that time to wait. And that's why we want to enhance their activities by providing them with more nutrition and oxygen, right? So whenever we do that, it will help us in uh, cleaning up this pollution faster, and that's why this is correct. Second statement: any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Now before I tell you whether this is correct or wrong, just let me highlight certain words. Any, readily, completely. What are these? Extreme words. And you know my feeling towards extreme words. So when we have these extreme words, I often told you that 9 out of 10 times they would be wrong statements. And here there are 3 of such words, right? So it makes it extremely difficult that these kind of statements will be correct. And that's why this is not a correct statement, because we are talking about microorganisms. And as I told you, that when it comes to heavy metals such as cadmium and lead, it's not easy for the microorganisms to break them down. All right. So yes, in certain cases, can they do it? Yes, they can. But in very extreme cases, only in certain cases that can be done. And that too, we have been trying genetic modification in microorganisms so that we can carry these kind of processes. So that's why it's a very extreme case, an exceptional case where we can even do it. In large number of cases, we'll try to use some other method, either some kind of industrial method, or if not, phytoextraction. So that's why in these kind of cases, instead of using microorganisms, we would prefer phytoextraction process. All right. So that's why we wouldn't consider statement number two as correct. Third statement is genetic engineering can be used to create microorganisms specifically designed for bioremediation. Can it be done? Of course, it can be done. We discussed about oil zapper, and we discussed that oil zapper is a formulation made of five different bacteria. And even in case of microorganisms, the second statement also, what I just told you is that we are trying genetic modification so that we can help better in bioremediation processes. So that's why statement number one and statement number three are correct, and correct answer will be C. Another question, a very simple one. Here I need much more number of. You've read it? Yes. The oil zapper was in the news. What is it? First statement says it is an eco-friendly technique for remediation of oily sludge and oil spills. Precisely what we just discussed. That oil zapper is a formulation of five different bacteria used for oil spills or used against oil spills. Correct. In bioaugmentation, also I discussed this. Yesterday also I discussed this. So you have oil spill here in A. So looks like a correct answer. It is the latest technology developed for undersea oil exploration. It is not related to exploration. It is for decomposition process, right? For bioremediation process. So B cannot be correct. It is a genetically engineered high biofuel yielding maize variety. I don't know whether you have marked C and D only to mock me or something, but I don't know why you would mark C. I You really have to concentrate in the classroom. I have given this answer at least four times since yesterday, and after that somebody marking C is not acceptable. Sorry. It is the latest technology to control the accidentally caused flames. It's not about the flames, it's just about the oil spills, right? So that's why D can't be the correct answer. Correct answer is A. It's a very simple question. All right. So now moving on to some of the specific topics. So until now, we have discussed some of the branches and how in those branches we are making the use of biotechnology. But from here on, we are now entering some of the very specific topics with regards to biotechnology, where we'll discuss about what is cloning, what is stem cell, and what is stem cell therapy, and all these kind of topics are what we are going to discuss in this part of our lecture. So the very first topic here is cloning. Now, when we say cloning, cloning is a process where we are basically trying to create a replica. That is what we mean by clone, correct? Clone is an exact replica. So, in case of creating this exact replica, we are basically trying to replicate some kind of animal. This example right here that you see is the example of the very first cloning that was ever tried, and this was a sheep called as Dolly. So this was the very first successful cloned animal sheep called Dolly, and this was 1997. Now, cloning as a process, what we are saying is that we are creating an exact replica. Now, when we say creating an exact replica, what do we mean? I mean, you might have seen some films, etc., where you'll see that one person enters a box and then something happens, and there is another person who is exact replica of this person comes out of the box. Correct? So, this kind of a scenario perhaps you might have seen in many of the films where they try to create these clones. For example, if you belong to the Shaktiman era, you would know this, that this is how Shaktiman's clone was created. Or in many of the other films also, in many of the Hollywood films also, these kind of techniques actually have been shown. So in this process, unfortunately, the things are not that easy. There are no two boxes where you can just insert one animal in one box and another animal comes out of another box. Unfortunately, no. Here, what we do is, this is a process that we say is a kind of an asexual reproduction. This is a process of asexual reproduction where we are trying to create an exact replica of the single parent who has contributed to the genetic material. So why, first and foremost, when we talk about reproduction, we know that there are two types of reproduction. One is asexual reproduction and the second one is sexual reproduction. Now in case of asexual reproduction, there is only one single parent from where the birth of the offspring happens, whereas in case of sexual reproduction, there will be two parents, one will be male and one will be female, and then fertilization will happen between the male and the female cells, leading to the birth of a new offspring. So that is sexual. In case of asexual reproduction, there will be only one single parent, and the birth will happen from that particular parent itself. So, for example, a simple technique like cell division that is seen in many of the microorganisms. So that cell division technique is nothing but asexual reproduction, because what happens in cell division is that there is one unicellular organism in which you have that nucleus, and nucleus starts to divide into two nuclei, and as it divides into two nuclei centers, it will just divide into two cells with one nucleus each. So that is nothing but asexual reproduction. Similarly, there is the process of budding, in which we'll see that out of the individual there will be a small bud that will be formed, and this bud will start to grow, and it detaches from this organism or this individual, and that's the birth of the new individual. So now in these kind of techniques, in all these asexual techniques, what's happening is that all the genes that the offspring will have will be exactly the genes that the parent had, because there is no mixing of genes. In case of sexual reproduction, there will be mixing of genes, one coming from the male and one coming from, or one set coming from the male and one set coming from the uh, female, and that's why the child or the offspring will have a mix of characters from the mother's side and the father's side. So that's why that's different. In case of asexual reproduction, all the set of genes are coming from one single individual only, one single parent only. And that's why it will be an exact replica of the parent in terms of its character. So that is how cloning basically is carried out, where this technique of cloning also is very similar, where we take the cell of a particular individual and we try to create a replica of it and try to create a new individual out of it with the help of certain techniques. So the first time we were able to do this successfully was back in 1997. Now, although we got this success, the success has not been easy, because compared to the plant genes, where we have been able to produce transgenic plants, a lot of hybrid varieties and all these things, doing the same in animals is not that easy, because the genetic structure of the animals are very different and very complex compared to the plant structures, and that's why we see that it is a lot more difficult process as compared to any such genetic engineering process that we have been able to carry in agricultural field or in plants in general. So that's why, compared to that, it has been very difficult. And even this success, just a trivia, do you know how many attempts it took for this success to happen? In how many attempts they were able to be successful? Okay, one correct answer from Akansha. Attempts for us to be successful. 277 attempts. How many attempts do you get towards your civil services examination? I'll not go there. So so it took 277 attempts, and then finally they were able to be successful. So that's why when we say first successful, you have to understand that the road was not easy. It was not easy to get to that success. And even after getting this success, the road even after that hasn't been easy, because still the process has been very complex and very unstable. So for example, let's say when we take the Indian examples of animal cloning that has been done. So the first example of animal cloning in India was of Samrupa, and this was the first cloned buffalo in the world in 2009. But unfortunately, the calf did not live for very long. It was just 5 days after the birth of this calf that it succumbed to some kind of lung infection, and that's why it did not live for very long. Then the second case was of Garima, and this was again tried, and in the same year, but again it also did not live for very long, lived for almost like 2 years, and then because of heart failure, it also lost its life. And then after that, there was CIRB Gaurav in 2016. In this case, we see that CIRB Gaurav was able to survive for longer as compared to the first two cases, and that's why there are other cloning cases also that have been carried on since. So for example, the next was carried out in 2020. This cloning was carried out in 2020, and here we see that there were two cloning cases that were carried on. One, seven clones of M29 Murrah bull, and then another one of the same CIRB Gaurav or Hisar Gaurav. A recloning was done. So this was the case, a case of cloning and recloning in the case, in India in back in 2020, and then after that there has been a new cloning that has been done in 2023, and this is a new cloning that was carried out this year, in the early months of this year. Now this time the cloning that has been done of a particular variant of cow called as the gir cow. Now, in this particular scenario, when it comes to this cloning, first time what we have done is that a cloning of indigenous cow variety has been done. Until now, the species for which cloning has been tried or hybridization has been tried, in all these cases, you'll see that those all have been foreign species, all right, not Indian species. For example, you might have heard of the Jersey cow. So for foreign species we have tried, but for Indian species we haven't. For the first time here we see that for Indian species also we have tried. Now the reason why Indian species, because Indian species are better. It was said that because Indian species are better suited to the tropical conditions. India's tropical climate. Hence, we have higher chances of survival. Right, higher chances of survival also, and immunity against diseases. So that was the basic reason why this time we thought that instead of looking at cloning of foreign variety of cows, let's also try goes, and if we are able to draw all the benefits that we are theoretically thinking of. So that's why we see that all these cloning techniques have been tried in India also. It has not been as uncommon as it used to be. Also, gradually we have been able to stabilize it better compared to the first few cases that we had in the world and in India. Compared to that, now we see that we have been able to stabilize the process much more, and hence we have a better success rate of survival with regards to all these cloning activities that are being carried out. So that is basically what we are doing with regards to cloning cases in India. Now, how exactly do we do cloning? What is this process of cloning that we do? So let's try to understand with the help of this diagram. So first and foremost, when it comes to cloning, we'll first have to isolate certain cells which are called as somatic cells. All right. First, we have to isolate the somatic cells. Now, what are these somatic cells? Somatic cells are all the non-reproductive cells in our body. For example, in humans, there are two reproductive cells. One called as the sperm cells, which are found in males, and second, you have the egg cells, which are found in females. So, apart from the sperm cells and the egg cells, all the other cells in our body, they are the non-reproductive cells, and these cells are called as somatic. All right, these are somatic cells. So, first and foremost, we will isolate somatic cells, and here we have shown a human. It need not necessarily be a human, it can be an animal. So first you have to isolate somatic cell from the person, or let's say from the animal. And from this somatic cell we'll first take out the nucleus. All right, we'll isolate the somatic nucleus, and once this somatic nucleus has been isolated, this will be inserted inside an enucleated egg, where you have basically removed the nucleus from the egg cell. And after removing the nucleus of the egg cell, we are inserting this somatic nucleus. So now you will have the egg cell with somatic nucleus. All right, we'll have the egg cell with somatic nucleus. So understand the value of this process, that why are we replacing this nucleus. What you know about nucleus is that nucleus contains the genetic material, right? All the genes, all the DNA, they are present where? They are present inside the nucleus, correct? So that's why if nucleus is retained, if you retain the nucleus of the egg cell, it simply means that the characteristics in that animal or in that cloned animal will be of the egg cell nucleus, correct? So we don't want that. So that's why we'll take away the nucleus from the egg cell, and that will be replaced with the somatic nucleus, because then all the properties that this clone will have will be of this particular somatic nucleus. All right, so that's why now you will have this egg cell with somatic nucleus. And once we have that, activation with the help of shock and chemicals will be done. With the help of certain chemicals and electric shock, we will basically try to activate it. Basically, this is something that will produce a bunch of cells together, which is also called as blastocyst. This is called as blastocyst. So we'll produce this blastocyst, which is nothing but a bunch of almost 150 undifferentiated cells that will be produced here. Now, after this, there are two ways in which it can be used. Either it will be used for the birth of a new cloned animal, then in that case, it will be called as reproductive cloning. If not, the same cells can also be used for some of the medical therapies. For example, in stem cell therapy, also something like this can be used. So that's why, if let's say we are using it for stem cell therapy, then we'll say that it is therapeutic cloning. And if we just want the birth of a new individual or a new cloned animal from here, it will be called as reproductive cloning. So in this manner, we see that this entire process of cloning will be carried on. So first and foremost, it will be provided with the help of somatic cells. So in from the somatic cells, the nucleus will be transferred into the enucleated egg cell, meaning the egg cell whose nucleus has been taken away. And then after that, it will be simulated with the help of electric shock and chemicals. This is where cell division in the cell will start, and we'll see that a ball of mass having almost around 150 undifferentiated cells will be produced. This stage is called as blastocyst, right? This stage is called as blastocyst, right? This is called as blastocyst. And from there on, we see that if let's say we need reproductive cloning, then in that case it will be inserted inside the uterus of a surrogate mother. And if at all, let's say if we need to just use these cells for some kind of medical therapy, then it will be taken to the lab, and from there on it will be used. So in this manner, we see that cloning as a process would be used. So in reproductive cloning, what we see is that the new created embryo is placed back into the uterus, which will take its time for the birth of a new individual to have happen. And in case of therapeutic cloning, we see that we'll try to harvest stem cells, etc., from here for medical purposes. So in this manner, the last step, there are two possible processes, whereas all the other processes before that, all the other steps before that will be same. So this is a question that was asked in your paper, a very, very simple question that was asked. Given the technology. So somatic cell nuclear transfer is being done in what process? In cloning, correct? So reproductive cloning of animals is where we are using this process. So correct answer will be C. So this is basically what we are doing. That first we'll take the somatic cell. From the somatic cell, we take away the nucleus. Why take away the nucleus? Why isolate the nucleus? Because nucleus has all the specific properties. So that's why nucleus, right? So why nucleus? I'm just writing it here so that it will help in your revision. Nucleus replacement done. Right. So because nucleus contains all the specific properties of an individual, and that is why it is so important that we isolate the nucleus, and it will be then produce. It will be inserted inside an enucleated cell. Again, cell division will start from here with the help of electric shock and chemicals, and then lastly, either it will be used for reproductive cloning or it will be used for therapeutic cloning. So this is how the process of cloning works. Now, one thing that we also need to understand is that there are certain concerns also that sorry there are certain concerns also that have been raised with regards to cloning. Now, cloning as a process is being used in animals, but in humans we haven't allowed the use of cloning. Governments all across the world have banned the use of cloning until there is a very extreme requirement where cloning as a process could be used. Otherwise, it has been completely banned. Now, why? Because there are certain concerns, and these are not technological concerns, technical concerns, but rather societal concerns that we have with the process of cloning. And that's why we see that these kind of restrictions or bans, etc., have been put over human cloning. So first and foremost, why? Because it might undermine society's respect for human life. Because clones, we if we now understand how to produce clones, we might not have the same respect for the human life as a society as we do right now. So these kind of things can actually change the complete structure of the societal thinking. So that's why this can be very dangerous from that perspective. Similarly, it can also have its effect on institutions like raising a family or even marriage, because then the individuals would know that I don't need to get married to have a child. I know that I can use the method of cloning and get a child with the help of reproductive cloning. So I don't need a child, so why to bother to even get married and do all that? So that's why we, in this particular scenario, these kind of situations might so happen that all these can lead to a very different scenario altogether. uh, and and of course, as a society, this completely changes the very thread of that binds the society. It might so look that you don't need family, you don't need to get married and all that, but these are only the things that you realize later how exactly it is changing the psyche and changing the entire societal structure, and it can go in a very negative direction, and that's why this is not something that is good. Similarly, we even think that it can become a global security concern. And why are we saying that it can become global security concern? Because let's say today some, let's say, terrorist organizations or even governments across the world might start creating cloned armies, think of cloned armies and start the cloning process, and maybe 15 years later they'll be ready with a cloned army. And who says that that can't happen? It can, right? And all these kind of scenarios can actually create global security concerns, because there, once you are creating those cloned armies, you'll not have the same respect for their life. You basically will think of them like you're creating machines or robots who are going to fight battles for you. Then there is very interesting concept which is called as designer babies. Now the concept of designer babies is when you're trying to design a baby. Now how are we doing that? We basically are, let's say what may happen is that in a child you want genes of certain person. You want your child to be a great mathematician or a great scientist, and for that you're just looking for somebody who has those genes, and then you would want the replica of that, those genes, to be your child, and uh, secure, you can say, your old age by doing that. So you might perhaps want that, and it's not something which is a science fiction concept. It is something that is possible even today, and there are processes that can make that happen even today. So that's why we don't want that to happen. That, and we'll discuss more about it when we talk about certain concerns in DNA fingerprinting also, but we know that these kind of situations are actually possible. Then at the same time, what we understand is that we know that experiments and research that we do on cloning, it also requires your work on embryos. Many a times you're working on embryos, and effectively when you're just doing that research or just doing studies, you might kill those embryos in the process. So a lot of embryos will get killed because of that, and in a manner it is equivalent to killing life once the conception has started. So many cultures don't allow that. Many of the cultures across the world, they don't see that in a positive life, positive way, and for them life begins at conception and not only after the birth of the child. So from that perspective also, there are ethical and moral concerns that are also raised against human cloning. So that's why largely you'll see that, in although in animal cloning there are not very strict extreme restrictions, but in case of human cloning there is. Then also to understand and not to confuse the difference between reproductive cloning versus surrogacy. Surrogacy is something that you also call as test tube baby and in vitro fertilization. So how is that process different? We know that in case of reproductive cloning, you are using the cells from one single parent and you are trying to replicate that. In case of surrogacy, that's not the case. In case of surrogacy or in vitro fertilization or the birth of test tube baby, in that still there are two parents who are involved, and fertilization still happens between the male and the female cell. So that's why it is sexual reproduction. Whereas in case of cloning, we know that it is asexual reproduction. So that's why as a process, it is completely different. At the same time, the uniqueness of the progeny, the uniqueness of the offspring. We know that the cloned baby, if at all, would be an exact replica of the parent. Whereas in case of surrogacy, the mixing of genes still has happened between the male and the female, and hence it is the progeny will be unique because it will be a unique set of genes. Then the problem with the lifespan. We discussed about the factor that lifespan of the cloned animals hasn't been that great. Correct? They have lost lives early. That's not a problem with surrogacy at all. The children who are born out of surrogacy, they have led a normal life. There haven't been any complications there. So that's why it is a much safer process, a much more secured process as compared to cloning. And then lastly, the ethical issues. We discussed about the ethical issues with the process of cloning. The ethical issues with regards to with regards to in vitro fertilization still aren't challenged as much. There are laws in place, there are regulations in place that take care of how exactly the surrogacy process has to has to happen. So that's why there are all these differences that we have between surrogacy and cloning. Now, before we move on to stem cells, if you have any doubts, please let me know. If you have any questions. of egg is not removed, and neither the nucleus of somatic cell is inserted, only nucleated egg cell is given electric shock, then in that case, the cell will retain all the properties of the egg cell. That's the only thing that will happen. So it's all about, just remember that it's all about uh, basically what nucleus you have. Where are you taking the nucleus from? That's it. So wherever you have the nucleus from, it will retain the properties of that individual. IVF, can you please repeat? So, in case of IVF or in vitro fertilization, the fertilization basically takes place uh, not in the original mother, but instead there will be a surrogate mother which will take care of the fertilization process. So, uh, the fertilization between the egg cell and the sperm cell will happen outside the uterus, and then after that the embryonic development that happens, that will happen in a surrogate mother, a third person, instead of the original mother. So that's the basic difference that's the change in the process that happens. And many a times, because of some kind of complication in the original mother, it might not be possible for the embryonic development to happen in the mother, and that is where the fertilization between the original mother and the original father will happen outside the uterus, and then after that the embryonic development will happen inside the third person. So that is what surrogacy is. Stem cells from umbilical cord are used in cancer therapy. Yes, it is. We'll just discuss about this in stem cells. Cloning word can be used for both humans and animals. Yes, of course we can. Just that it is allowed in animals mode and it's not allowed in humans. Couldn't make the running notes because the slides were running fast. I've given you the entire stories, and actually everything is there on the PPT. Everything that I said, uh, the summary of everything is already there on the PPT, so don't worry. Please explain the lifespan again. So what I was saying about the lifespan is that in case of lifespan for a clone, we have seen the examples in case of India also, that cloned animals did not survive for very long, right? They did not survive for very long. Whereas in case of surrogacy, that hasn't been the problem. So that's why the problem with lifespan, which is questioned in terms of cloned animals, is not questioned when it comes to uh, surrogacy. We saved the Einstein brain. Okay, any secret cloning, if if at all it is, as you have mentioned, it is a secret, so nobody knows whether it has happened. Recently, we saw in the news dead body of alien, still to be confirmed. The one I think you are talking about, the Mexico case, still to be confirmed what exactly it is. Didn't understand the difference between saturated and unsaturated region in bioremediation. So basically what I'm saying is that saturated, when do you say something is saturated? When it cannot absorb more. So when, for example, even in clouds, when do we say that it rains? When the clouds become saturated with water, correct? And then it rains. So saturated means that it cannot retain more of moisture. In this case, it cannot retain any more moisture. So it will be said that it is saturated zone because it is near the water table. So uh, its capacity to absorb more moisture is very limited. Whereas in case of the top layer, which is the top layer of the soil, the moisture retaining capacity is still there because it is not near water anyway. And in you also have the sunlight that also leads to it having less moisture. How will the egg come from? It will come from a female body, a female cell. Can't get that human body slide. Let me just repeat it once again. I'm saying that we first take the somatic cell out of the body. Somatic cell is any non-reproductive cell in the body. From there, nucleus will be taken away, right? Nucleus will be taken, and this nucleus of the somatic cell will be inserted inside an enucleated cell. Enucleated meaning the cell that does not have nucleus. So inside that, because we have already removed the nucleus of this egg cell, and then we insert this nucleus inside this enucleated egg cell, and then after that, with the help of electric shock and chemicals, we will produce a blastocyst, which is a bunch of undifferentiated cells. And from there on, either these cells will be inserted inside a female body, where inside the uterus the birth of embryo, or rather the development of embryo, will take place, or it will be taken to a lab from where it can be used to harvest stem cells. In surrogacy, we have the nucleus of both the parents. Yes, yes, we do, because it's a normal reproduction process. Oil zapper. So oil zapper, basically just understand that it's a hybrid of various different types of bacteria, especially five different types of bacteria. And this formulation is used whenever oil spills happen. So wherever oil spills have happened, we basically dump these bacterial formulation there, and these bacteria will now start to eat upon all the oil spills, all the hydrocarbons that have spilled on the sea or on the ocean, and they will just eat up, and they will decompose that hydrocarbon into carbon dioxide and moisture, and hence they'll be able to take care of the hydrocarbon spill that has happened as oil. In therapeutic cloning, after the cell division and embryo development, what will they do? They'll basically use it for stem cell production, and we'll just discuss that. Just wait. The nucleus of the gamete of the egg cell is also haploid, so it won't result into an organism. So somatic nucleus is needed. Yes, you can say that. Do cloned animals are unfit as compared to non-clones? Saying unfit is not the correct word, but rather we'll say that the process is unstable. So that's why, because the natural immunity that they otherwise get through a natural process, they don't get that. So that's why we have to keep them under some special conditions, away from the outside world, little bit isolation as compared to others, so that we can enhance their lifespan. Do we have to make running notes? No, you don't have to, because anyway, whatever is required, it's there on the PPT already. Either it is written or I write it. Whatever is extra, we do write it. So that's why you don't have to worry about making notes. But if you are in a habit of making note, then it's fine. You can keep making it along with the class. Does it mean that if we want to clone the female, we did not need the nucleus of? No, 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 no. Because we still need the somatic nucleus. Remember this, that you don't need the egg cell nucleus, you need the somatic nucleus. So even if it is a female clone, even then you'll have to use the somatic nucleus of that female. Can you please show the buffalo slide that you wrote? Okay. This was, yeah, here. In cloning, can we insert the cloned embryo into the same mother animal's uterus instead of surrogate mother? Yes, we can. Why not? What is different in PPT and annotations? There is no difference. PPTs are annotated once we are done with the session. If the student wants to give its attempt in 2025, is it sufficient along with current affairs? Yes, of course it is. Absolutely it is. Second question I did not understand. Second question. I don't know which question you are talking about. If you can mention the question. So I think I've taken many of these are repeated questions. What is genetically identical? Same as parents? Yes, exactly. Where we are using against oil spills. That's it. Hmm. So let's move on. There are some questions pending, but I'll take it after the break again. Huh. So the next topic that we have is stem cells. Now, with regards to stem cells, first and foremost, try to understand what do we mean by stem cells. So stem cells are undifferentiated cells. They are cells that do not have any specific function. Now, if you look at all these are the different types of specialized cells in the body. All right, these are specialized cells in the body which have one kind of function or the other in the body. So for example, we have the red blood cells, we have the white blood cells, we have the nerve cells, brain cells, heart cells, skin cells. There are n number of different types of cells in our body. And what you'll notice is that all these cells, they also look different. All these cells, they also look different, and they look different mainly because of the fact that they have their different functions, and according to the required functions they will have their shapes. For example, the red blood cells will be round in shape. Now why they are round in shape? Because they have to travel inside the cells, and for them to travel they'll have to have that shape that makes it easier for them to travel. Similarly, the nerve cells will be very long and pointy cells. Why are they long and pointy? Because they have to transmit the information from one end to another. So it makes it easier for them to transmit this information by being so. So that's why you'll see that all the different cells have their specific functions, and their shapes are also according to the required functions. All right. Now, when we say stem cells are undifferentiated, unspecialized cells, meaning that these are cells which do not have a specific function in the body, but they are the origin of each and every cell in our body. Each and every cell in our body, they basically come from stem cells. So that's why stem cells, you can say in a manner, they are the raw material. They are the raw material for every specialized cell in our body. Every specialized cell. And not only in humans, for every specialized cell. So, and not only in humans, but even in other animals, and even in plants, there also we see that even they have stem cells. So whether it is higher plants or higher animals or humans, we see that cells are found in all of them. So stem cells are raw material. These are the unspecialized cells which will grow into a specialized cell. So whether it is a heart cell, skin cell, blood cell, red blood cell or white blood cell, nerve cell, muscle cell, etc., they'll all be coming from stem cells. All the origin of all these different cells, the origin is the stem cell. So that's why stem cell becomes crucially important for the growth and development of each and every one of them. And because these are the cells from where they all develop, that is what makes stem cells very easy. So that's why stem cells can become anything. Stem cells have the property that they can develop into any kind of cell. All right, so that's why they can become anything that they want. So that's the beauty of stem cells. That's the, you can say, importance of the stem cells, that stem cells are the raw material for all the other body cells. They are the raw material for all the other body cells, and they basically will grow into these specialized functions. So that's why they'll have the capacity to just become anything. They can just grow into any kind of cell, but with certain restrictions that automatically comes in some of these cells, and we'll discuss about that as well. So that's why these are the unspecialized cells which have not yet developed, which have not yet developed into mature specialized cells. Now, when it comes to stem cells, today we see that we have also found a lot of use for this stem cell, right? We have found a lot of use for all these stem cells. Now, what exactly are these uses, and how have we come to these conclusions, right? How have we come to these conclusions? So there are two important properties. There are two important properties of stem cells: ability of self renewal and ability to specialize. So what are these two properties? Let's try to understand, because it's these two properties that makes them so special. So first property is their ability of self renewal. Now, ability of self renewal means that they can self renew, meaning that they can create multiple copies of themselves. Meaning, before they start to specialize, before the stem cells start to specialize, there will be multiple cycles of cell division. And in cell division, we discussed that cell division is an asexual process. So whenever cell division happens, it's just the replica of cells that are being created. So that's why for us to work with, there are multiple stem cells available. So their ability to self renew becomes very, very crucial. So they will go undergo numerous cycles of cell division while maintaining the undifferentiated state. So there will be multiple cycles of cell division before they start to specialize. All right. So basically, we are saying multiple cell division cycles before they start to specialize. All right, multiple cell division cycles before they start to specialize. So that is basically what we call as the ability to self renew. Second is their ability to specialize, because what we know is that the same stem cell can grow into a brain cell, can grow into a heart cell, can grow into a muscle cell, can grow into a skin cell. So that's why their ability to specialize is very, very unique, and that is what makes them very, very useful in medical procedures. We'll discuss about that, how exactly that happens. We'll discuss about that. But we see that their capacity to differentiate into specialized cells becomes very, very crucial, and that's why they are so important for us, because they can specialize into various different types of cells. They can specialize into various different types of cells, and because of that they will have very special applications. So first and foremost, we'll try to understand where exactly do we get them from, and that's based on that we see that there are multiple different types of cells. So we have three types of stem cells called as totipotent cells, pluripotent cells, and multipotent cells. Right? Totipotent, pluripotent, and multipotent. Now, what is totipotent? Toti comes from, you can say in a manner, comes from the word total. All right, it comes from the word total. So totipotent. Pluripotent comes from the word plural, and multipotent comes from the word multiple. So what does this mean? Totipotent cells are the cells that have the capacity or the capability to specialize into any cell without exception. Without exception, totipotent cells can just specialize into any kind of cell. Now, these totipotent cells are very special and very rare. They are found, they can only be derived within the first two cell division cycles after fertilization. Just after when fertilization has happened, within the first two cell division cycles, that is where we can derive the totipotent stem cells from. After that, we can't get totipotent cells anymore. After the first two cycles of cell division has happened, after that you can't get totipotent cells. After that, what you can get is only pluripotent cells. All right. So totipotent cells become very special. Why? Because they can specialize into all cell types. Essentially, we are saying they can specialize into any cell. All right, they can specialize into any cell. So that's why these are the specific stem cells that can specialize into any kind of cell. So totipotent cells or totipotent stem cells can specialize into any kind of cell in the body. Now, the stem cells that we derive after the first two cycles of cell division, they are called as pluripotent. So they will also have a very good capacity to specialize. They can specialize into, I'll write, almost any cell. Right? They can specialize into almost any cell, but not all the cells. Especially the reproductive cells, they cannot specialize into reproductive cells. So pluripotent cells will be able to specialize into, sorry, I missed here, almost all the cell types. Now just remember that both of these are embryonic cells. Both of these are embryonic cells. They are derived from embryos. Derived from embryos, and they are also derived from embryos. So both of them are being derived from the same place, just that totipotent stem cells are coming from within the first two cycles after cell division, and pluripotent cells are coming after the first two cycles of cell division. So that's the basic difference between the pluripotent cells and the totipotent cells. So after this, we have the multipotent cells. Now, multipotent cells are not derived from the embryos. They are derived, we say that these are derived from the adult stem cells. Now, when we say adult stem cells, we do not really mean that these are adults, that you are only deriving it after 16 years or 18 years of age of somebody. Adult means that the birth of child has happened or birth of the animal has happened. So after birth, so adult stem cells mean stem cells derived after the birth. stem cells derived after the birth. So that is called as adult stem cell. Now, in case of adult stem cell, we see that it has a limited ability to specialize. All right, limited ability to specialize. So it has a limited ability to specialize, meaning that adult stem cells or the multipotent stem cells, they cannot specialize into anything, which was the case with totipotent or pluripotent cells. Because here already the body has developed, the individual has developed. So now what will happen is that the stem cells which are near the heart, only that will specialize into heart cells, or let's say muscle cells near the heart. The stem cells that are near the brain, that will specialize into brain cells. So it will become more limited in its ability to specialize compared to the totipotent and the pluripotent cells, which have a much wider ability to specialize. So that's the basic difference between these three types of stem cells. All right. So not only adult stem cells, but even the ones that are derived from the umbilical cord of the blood cells, etc., even they are multipotent cells. They are also called as multipotent cells. So in all these cases, just after the birth also, adult stem cells will be formed, and ultimately they will have limited ability to specialize, because now all the organs of the body has developed and all these developments in the body have happened. So that's when now the stem cells will have a limited ability, and it will purely depend on where are you deriving these cells from, right? So that's what we observe here. Now, coming to the use of stem cells, what we understand is that there are various different medical applications for which stem cells can be used. So, for example, for regenerative medicine. Now, in case of regenerative medicines, basically we try to regenerate damaged tissues, regenerate damaged cells or tissues. In that particular scenario, we say that it is regenerative medicine. So, for example, repairing the heart muscles which might have happened because of heart attack, or even in case of eye surgeries, we see that something like this is used, or even for burn victims or injuries, we see that skin regeneration can also be done. So all these things can be done with the help of stem cells. Why exactly we see that? Because we know that the stem cells can grow into a new cell. So whatever damaged skin cells or damaged heart muscles, etc., we have, we will try to replace that with a stem cell, and this stem cell will now grow into a normal cell, into instead of the damaged cell, the stem cell will now grow into a normal cell. So that is basically what we'll see with regards to the regenerative medicine. Similarly, it can also be used for drug testing. It can also be used for drug testing. Now, in case of drug testing, basically what we do is that first we will try to mimic, or let's say we'll try to grow a tissue or an organ from the stem cell. All right, we'll try to grow a tissue or an organ with the help of a stem cell, from a stem cell, and we'll do all the testing of the drug on that tissue or on that uh, cell, or that organ. So instead of doing these tests on a person, we can do all these drug tests, or even for that matter, even for the vaccines, the testing can be done on stem cells, or rather the created tissues or created organs out of the stem cells. So there the drug testing can be done, and that's why it will be safer, at least. Otherwise you would have done it on a person. So so that's why the safety and the efficacy of new drugs, and also for vaccines, for drugs or for vaccines, we can make the use of stem cells. So basically tested on organs or tissues grown from stem cells. All right, so that is basically what we'll try to do here. That first, let's say there is a new vaccine. All right, there is a new vaccine that is supposed to work on the lungs of a person. So then in that case, instead of testing on a person, which can also have a lot of side effects, and even in cases it can even lead to death of a person, so instead of taking that risk, what we'll try to do is we'll try to grow these organs like lungs, etc., or grow the tissues which are near the lungs, and then do all the tests there and see what's happening. How is the lung responding? How is this artificial lung responding? And if, let's say, if it is responding well and we see that there are no side effects and there is no harmful effects here, then in that case we can use the same on people also. So that's why it makes the use of these drugs and new vaccines much safer. Then it can also be used in disease modeling. So what we can basically do is that we can create models of genetic diseases or models of these diseases and see how exactly is the growth happening. Because ultimately, see, what happens is that maybe some cells are already affected because of some kind of disease. Some cells, some tissues, some organs are already affected. So what we'll try to see is we'll see how exactly the normal growth or how exactly the diseased growth is happening inside that person versus how a normal growth happens, and what is the step where some kind of problem is arising in this stem cell, or the growth that is happening in the person inside that stem cell. So that will give us an idea about what's going wrong, and then we can try to treat the disease by understanding what's happening there. Then we have stem cell therapy, in which we see that conditions like leukemia, lymphoma, and blood or immune disorders can actually be solved with the help of stem cell therapy. There have been cases where we have seen that we have been able to cure cases of cancer or even cases of AIDS with the help of stem cell therapy. Although they have been very small in number, where we have actually been able to get all these benefits, or we have been able to be successful, but we have seen success. And yes, in the future we expect that the success rates will be higher. Of course, these methods are very, very difficult. They are very, very complex. And that's why until now, uh, the success that we have seen is very limited. But at least we have tasted success with the help of stem cell therapy. And that's why in the future, we believe that it will be much better as compared to where we are right now. Then after that, it can obviously also be used in a lot of basic research. It can be used in multiple different ways, and that is basically what we are looking towards when it comes to the use of stem cell therapy, that in various medical uses it can actually become useful. Similarly, it can also be used in tissue engineering. It can also be used in tissue engineering. So tissue engineering basically is a method where we try to create new tissues or try to create new organs to replace a diseased or an infected organ, or maybe even because of accidents, etc. So for example, let's say due to accident somebody has lost something, or some organ has been damaged, or some part has been lost. So in those kind of cases, tissue engineering can be used to repair the damage. And also in we see that in neurodegenerative diseases also it can be useful. Diseases such as Parkinson's or Alzheimer's diseases, in these kind of cases also it can be very useful. So in all these cases we do see the use of stem cells, although success until now has been very limited. Success has been very limited, but at least we have seen the success. And that's why we see that a lot of investments are being made, a lot of new methods are being developed, a lot of research and development and studies keep on happening as we speak. And countries all across the world have been trying to get a better success rates in case of stem cells and the various ways in which the medical science is trying to use stem cells. Now, one last thing about the issues related to stem cells. So, for example, there is the ethical issue, and the ethical issue largely is in the use of the embryonic stem cells. With the this basically is arriving when it comes to the totipotent or the pluripotent cells. Totipotent or the pluripotent cells, because this is where the work is happening on the embryos, and we really don't want to use the embryos because it has ethical concerns. What if tomorrow commercialization of embryos start, because just because it will be sold well in the market, because research has to happen on embryos. So that's why all these are possible scenarios which we need to be aware of, and hence we see that there are all these ethical issues with regards to the stem cell, with regards to especially the totipotent or the pluripotent cells. The other side of the story is the use of adult stem cells, where we see that they have a limited ability to specialize. So, on one hand, we don't want to use pluripotent or totipotent cells, because there it needs work on embryos. On the other hand, we know that adult stem cells are not as useful as the pluripotent or the totipotent cells have been. So, that's why there is a problem here. So that is where we see that this is a problem with the multipotent cells, that they have a limited ability to specialize. And now there is a way out, something which is called as induced pluripotent. Induced pluripotent stem cell. Now this is a kind of stem cell where we try to convert an adult stem cell into an embryonic stem cell. We try to convert an adult stem cell to embryonic stem cell. So this is something that actually is now possible, that we can convert adult stem cell into embryonic stem cells. So in that case, all these two issues that we are discussing here, it is a solution to both of these issues, right? It's a solution to both of these issues, and that is where we see that conversion of adult stem cells into embryonic stem cells, which is called as induced pluripotent cell, will be very, very useful. So these are some of the things related to the stem cells. Look at this question. This was asked in your paper. Please mark your answers. Yeah, a lot of you have given the correct answer. Wonderful. So almost 75% of you have given the correct answer, which is B. So question says, with reference to stem cells frequently in the news, which of the following statements is or are correct? First, stem cells can be derived from mammals only. First and foremost, this term only. But what we discussed is that it's not only mammals, but we know that all the higher animals, plants, and humans. Although humans also are nothing but higher animals, but we try to keep ourselves aside from higher animals. So all of these have stem cells. In case of plants, basically you have cells which are called as meristems. Meristems or meristematic tissues, which are, you can say, the tips of uh, leaves or the barks. So wherever you have the tips, wherever new growth is happening, wherever from new growth is happening, that's the part that has stem cells in plants. So that's why not mammals only, everywhere. I mean, all the higher plants and animals. Stem cells can be used for screening new drugs. Absolutely correct. We discussed that drug testing can be done with the help of stem cells, that we grow new organs or new tissues, and that we do. Stem cells can be used for medical therapies. Absolutely correct. That is what stem cell therapy is. So 2 and 3. Correct answer will be B. That is 2 and 3 only. Let's take a break here. Uh, right now the time is 6:40. Is when we are trying to replicate a disease. We are trying to understand when exactly is the disease developing in an individual, especially when you have a genetic disease. So genetic disease means that it's there in the body of the individual already. So when the cells are growing out of the stem cells, while developing, they are growing the disease. So at what step are they developing that disease? We try to observe that versus how a normal stem cell grows into this specialized cell, and that will give us an idea about how exactly the disease develops, and maybe we can uh, make some mechanism against it so that we can prevent that disease. So that's what happens in disease modeling. Difference between sexual and asexual reproduction, simple, that in asexual reproduction you have only one parent, in sexual you have two parents, and a mixing of genes happens between male and the female in sexual reproduction. Three parents, maybe we'll discuss that. We have not yet discussed it. How much time after birth is multipotent? Just after birth. Difference between pluripotent and multipotent. So as I told you, pluripotent is in the embryonic stage, when the growth of the progeny hasn't happened yet. It's in the embryo stage. And multipotent is when the growth has already happened and all the parts of the body, organs, etc. have already developed. So that is what multipotent is. In vitro fertilization or IVF examples of cloning? No, I told you that there's a complete difference between the two. IVF is the process of surrogacy, which is very, very different. Please explain the third type of stem cell. Third type of stem cell basically is multipotent cell. Multipotent cell is the stem cell, for example, the ones that are found in us after the growth has happened, after the birth of the child has happened. Then the kinds of stem cells which are found, they are called as multipotent. Please explain tissue engineering. Tissue engineering is when we are trying to repair a tissue. All right, maybe because of an injury, something has happened, or for the burn victims, etc. When we are trying to regenerate those tissues which have been damaged, that is called as tissue engineering. Can organs other than liver rejuvenate themselves? Not really, they can't. Liver has that property, but most of the other organs, they don't. What is the problem with adult stem cells? That they're limited, that they can't really grow into anything. They'll grow into only the cells which are near the growth site of that stem cell. Disadvantage of multipotent, the same thing what I just discussed. Stem cells are continuously developing in the body. Yes, every second lakhs of stem cells will be developing in the body. Eggs also contain single parent nucleus. Why somatic cell is used? Somatic cell is used because you need the properties that are there in the particular somatic cell. So that's why you cannot do it with the help of egg cells only. And also there are other technical issues also which come there. Please explain how different embryos can be generated by stem cells. No, embryos are not being generated by the stem cells. Stem cells are being generated from the embryos. For any use application of stem cell, which cell is used, embryonic or stem cell? Largely, it is all stems, all adult stem cells only. How stem cells can repair the organ? They will simply grow into a new organ because they have that. That's what we are saying. That's the property that stem cells have. That they'll grow into the required cell by themselves. Is surrogate mother always necessary for cloning? No. Why stem cells are preserved by hospitals for children? Because they'll be using it for some kind of medical therapy later. Stem cell present all over the body? Yes, of course, because every cell is growing out of stem cells, so it's there everywhere. So bone marrow, for example, is the main site of stem cells. In lecture 3, space policy 2023, not attached with the PDF notes, please attach it. uh, I'll check with the subject matter experts. I'll ask them to do it. What is the use of cloning? There are various different purposes for which cloning is being used. Either it could be that better species or healthier species are being cloned so that they can be very useful for us, maybe in agriculture, maybe in poultry, maybe in the milk industry. Everywhere we'll see that they'll in the dairy industry, that is, we'll see that it will have their effect, I mean have their advantages everywhere. In humans also, do we have stem cells? I can't answer that. How is that even a question? That makes me doubt myself that after saying everything, if such a question is being asked. Yes, of course, humans have stem cells. Every, I told you, every animal, every higher. You just look at this particular screen right here. You can't ask this question. All right. So the next topic that we have is DNA fingerprinting. Now, in case of DNA fingerprinting, what we basically see is that this is where we are trying to identify the complete or partial set of genetic information of a particular individual. Now, fingerprint. When we use fingerprint or when we talk about fingerprint, what is the uniqueness of fingerprint? Why is fingerprint of a person important, or what is the uniqueness about the fingerprint? Yes, it's one of a kind, correct? It's unique. Every person's fingerprint is different, right? Nobody will have same, no two people will have same fingerprint. So when we say DNA fingerprint, something similar we see with our genetic structure also. The genes that I have will be different from the genes that you have. The genes that you have will be different from everyone else, even your parents. Your parents, with your siblings, with your children, the the stem cells that you have, the stem cells basically, sorry, the genetic structure, the genes that you have will be largely similar, but there will be differences. There will be differences. Even if you have twins, even if you have twins of the same gender, even then, even then there will be slight differences between their genetic structure. So that's why the genetic makeup of every one individual is different, and that's why it can be used for identification, to either establish a relation between the two or to also differentiate between the two. But the applications of DNA fingerprint goes just beyond just a basic identification. Now, in case of identification, when it comes to it, it can be used for identification of criminals. For example, if you see this picture right here, all right, if you look at this picture right here, you see that this is a crime scene sample that was collected from some kind of crime scene, right? And there are three suspects. Now, if I ask you, can you tell me, out of these three suspects that we have right here, which suspect's DNA structure matches with the crime scene sample? Yes, correct. Second one, right? So as you can see that this and this are similar, correct? Which is not similar in this case. this and this are similar, which is not similar in this case. This and this are similar, which is again not similar in both these cases. So we understand that the second suspect's genetic sample is exactly the same as what was collected from the crime scene. Then at least you know that the second suspect was at least present at the crime scene. You still will have to prove that the second suspect was actually the criminal, right? So identification of the criminal can be done with the DNA samples, and that's where DNA fingerprinting becomes useful here. Then it can also be used for paternity tests and even for claims over dead bodies, because that is where you can also establish relationships also, because a large amount of genes will be similar, and that will be able to strike the resemblance, and you'll be able to get the identification. Or for paternity test also, to uh, establish the paternity, we see that the same identification method can actually be useful. Then apart from that, in also making effective drugs. Now, how to make effective drugs with the help of DNA fingerprinting? If you remember, we discussed something called as pharmacogenomics, right? We had discussed pharmacogenomics yesterday. So pharmacogenomics, what we discussed is that when we try to make medicines according to the understanding of the genetic makeup of a person, something that will have a good positive impact on the genetic makeup of a person. So that is what pharmacogenomics is about, right? So that's why when it comes to pharmacogenomics, it becomes very, very useful in making effective drugs. So that's why with the understanding of the genetic structure, with the genetic makeup, the entire DNA structure of a person, will be able to make better drugs with regards to those genes. So that's why to make effective drugs, we can make the use of DNA fingerprinting. Similarly, in the treatment of genetic vulnerabilities as well, especially in the identification of genetic vulnerabilities beforehand, so that you can treat them early, you can identify them early. For that also, DNA fingerprinting becomes very, very useful, because we'll be able to identify certain genes, that if these genes are present in the parent, these are the genes that are responsible for the diseases, and these genes might also transfer to the child. So that's why there also it is a process that can be fairly useful. Then in wildlife management also, it will help us in better understanding of what are the external factors that are leading to the decline in population. That if there are certain environmental factors or geographical factors or even anthropogenic factors that are leading to the decline of the population uh, of a species, all those can also be identified in the genes itself, and we can take precautions, we can take steps accordingly. So that's why in wildlife management also, something like this becomes very, very useful. So there are multiple different uses that we find for DNA fingerprinting. But at the same time, we see that there are certain concerns with regards to DNA fingerprinting also. So, for example, the gene pool, the information about the gene pool can lead to a reference for designer babies. And that is something that can happen. That, let's say, what happens is that the genetic information about people or public in general is available. So then what happens is that we'll be always looking for those superior genes. We might want that in our children, the children should be born with superior genes. And we are free to define these superior genes in whatever way we want. So whether it is intelligence that you're looking for, whether it is good looks that you're looking for, whatever kind of, or whether it is somebody who is leading a very healthy life, and you just want that those genes should also be present in your child, and you'll try to introduce those genes in one way or the other. So that's why doing that is possible today, and hence that's why we see that these kind of things can become a concern, that we'll always be looking for those superior genes, and looking for introducing those genes in the babies, or in even before the birth of the child, something which is also called as germ line editing. Germline editing. So germline editing basically is genetic modification, genetic modification in the reproductive cells, which can lead to the genetic changes or rather carry on for multiple generations. So what I mean is that when germline editing is done, the changes that you are doing is permanent. All right, the changes that you are doing is permanent, and that's why these changes will keep on uh, traveling from one generation to another, to the third generation, to the fourth generation, and so on. Whereas in somatic modification that we do, it will only be for that individual where the somatic modification has been done. But when you do modification, the germline editing, whenever it is done, these kind of modifications will just carry on for generations. So that's not good. It's ethically also it is wrong, because you are taking a decision for somebody without asking them. So for example, you are looking for those genes that are related to superior mathematical aptitude, and you want those genes in your child. So you have not asked the child whether the child wanted to be a mathematician at the first place or not, or whether the child wanted to become a lawyer or go in the humanities sector, or maybe become a CA. But you did not even consider that, and you just wanted a good scientific gene in the child. So that's why all these things are possible, and these are also unethical, because you are doing something without the consent. You are taking the decision for the upcoming generations, and not only one single generation. So that's why this can be wrong. This can go in a very negative direction. Similarly, the genetic privacy of individuals can be violated. Now this is also a big challenge, because then what will happen is that you know about the genes of everyone, and you know what are superior genes and what are inferior genes, and we will find a new way to discriminate between people who have better genes, so called better genes, versus people who do not have those better genes. And we probably will see a shift in the societal uh, uh, vision, and we might see that people are being looked down upon because they do not have those superior genes. It's all a possibility in the future, right? So that's why this is also a concern. Similarly, genetic information can be misused for commercial purposes, right? So now, when it comes to misuse, what can happen is that, let's say if the pharmaceutical companies have the idea about your genes, they know about your genes, then what may happen is that they might use, or rather misuse, that information. They'll say that, okay, look, you have this and this gene. It also means that you are going to be susceptible to this disease and that disease and that disease and so on. So you will also be afraid. You'll think that, okay, fine, I mean, I don't know, but if that person is saying, it probably is correct. And they will start selling you medical packages. They'll start selling you medical packages based on whatever information that they have given you. You do not understand. In general, we do not understand science well. That is one of the problems with this class. So in general, science is something that they'll just try to I mean, just try to explain you, right? So for example, you look at so many advertisements, whether it is advertisements of the electronic products, or maybe advertisements of the cosmetic products and so on. You'll see they talk about so much technology, they talk about so much science that we don't understand. Just that when they say it, it looks good, correct? It sounds good. So we probably buy because of that. We buy a lot of things because of that. So in the mobile phones, they'll talk about n number of different types of technologies that you don't even have any idea about. You just know that it sounds good, so it must be good. Same goes with cosmetics as well. So ultimately this happens. For example, okay, one cosmetic example: charcoal. Charcoal in your toothpaste, charcoal in your soap, charcoal in your shampoo, charcoal in everything. So does it mean that I should just go and eat charcoal for my well-being? The answer is of course no, because if you do that, you'll be hospitalized the same day. So, but why charcoal? They'll say that charcoal has antioxidants. Okay, sounds good. So probably I should get them because I'll get antioxidants from there, and it must be good in some way for me. What you don't understand is that our body gets antioxidants from a lot of fruits and vegetables. So rather eat those fruits and vegetables, and you don't have to eat charcoal. You don't have to depend on charcoal for your antioxidants. But the advertisements will sell you antioxidants in the form of charcoal, and we also get mesmerized by these advertisements, and we go by that. Either that, or they'll try to instill fear in you. They'll say that if you don't do this, if you don't buy this, these are the problems that people are facing today. So you'll go and you buy that product. A very beautiful example here is of a recent advertisement that I saw about, I think, it was good night or some kind of mosquito repellent. So there's one information I'm sure many of you also know, that the mosquitoes that are responsible for dengue, they are also active during daytime, not only during evenings or night, but also during daytime. Information that many of us know. So now that advertisement is talking about that it's not that you have to use the mosquito coil or the all out or whatever they are giving you, that it's not that you have to use it only during night, because mosquitoes are also active during day. So now what happens? Their sales will increase, because now they have made you afraid of this disease and they can sell the product on that, right? So that's why we see that all this can be done, and that's why, because we don't understand, we have a very limited understanding of science. That's why science can be misused against you, right? Maybe all the explanations that I'm giving you, who knows which is what whether these explanations are correct or wrong. Just that you are hearing it for the first time, so you will, I'll not go there, right? So that's why this is also a big problem. Now also, genetic information might be misused for creating weapons of mass destruction, right? Weapons of mass destruction can be created, because here also what happens is that your genetic information might be used against you. Let's say if you take example of India. So we know that for India, the southern side, you have the peninsular part of the country, which is surrounded by seas and ocean. In the north, on one side you have the Himalayas, on the other side you also have the Karakoram and the Thar. So that also, in a manner, you can say, isolates us from the other parts of the continent, right? So genetic mixing has happened in this subcontinent for a very long time. So there are a lot of common genes. There are a lot of common genes across India, right? A lot of common genes across India. So if somebody had to target India, they'll have to just find that common gene. They'll have to find that common gene, and they can actually create a weapon of mass destruction against India. Forget about India. What we have seen with COVID 19, a virus against which the world did not have an answer. Our genes did not have an answer. Our immune system did not have an answer. And it led to so much of devastation all across the world. Right? We are still trying to recover from it. So what happened here was that there was this virus uh, against which we did not have an answer, and this is a possibility that something similar can be created. Are there not people who also claim that this was made in the lab? Whether it was made in the lab or not, we will never know, because it's China. So information is completely opaque from China, and you'll never know whether this was correct or this was actually the truth or not. But whether it was the truth or not, we know about the possibility that something like this is actually possible. So that's why it's all very dangerous. Then we also discussed about gene therapy. What we understood is that it's either about replacing the gene which is responsible for the problem, or inactivating or knocking out a gene, or maybe introducing a gene. So addition, subtraction, modification. These are the three things that we can do as gene therapy. Now, gene therapy is when we are using the required gene. Either we are introducing a new gene, or we are modifying the gene, or maybe we are just knocking off or inactivating a certain gene. So there are all these things that we can do in these kind of cases. So for example, when we knock out a gene, it is also called as gene silencing. This is also called as gene silencing. So because we are silencing a gene, we are trying to inactivate a gene. So all these kind of things are done in gene therapy, and with the help of gene therapy, we try to solve certain unsolvable problems, the diseases against which we do not have any answer today. We are trying to use methods like gene therapy or stem cell therapy for that particular purpose, right? So that is where we know that it will have a very, very important work. Now, when it comes to gene therapy, although we do see that there are a lot of advantages that we see, there are a lot of studies happening, there are a lot of new researches that are happening in the field, but we still see that it is not completely working for us today. There have been cases where it has been very, very successful, but that's very limited. So there have been concerns. Three important concerns that we have. First is the short lived nature of the treatment, that the treatment has not been widely successful. All right. Short lived nature, meaning that today it is showing some positive impact on the body, but that impact very quickly ceases to exist. What happens? The reason for something like this, the short lived nature, is that in our body itself, we see that uh, the body starts to go back to its previous ways in a manner. Let's say when you talk about the cell, what we know is that we have cells all over the body, and every cell in the body will keep on growing, develop, and die. And as the cell dies, it will be replaced with a new cell. And this is a process that keeps on happening every second in the body. And just to understand the sheer number of this and how this happens, by the time, just let's say, one sentence is completed by me, almost 10 lakh cells in my body will be replaced. So that's the speed in which we see that the growth and development of cells happen, and they keep on dying, and they are always replaced with new cells. So when gene therapy is introduced, what may happen is that because this functioning is so quick, whatever new genes have been introduced or whatever new methods have been introduced, the body might forget about it and go back to its original ways. And that's why many a times the gene therapy might not last for very long, and anyone who's going through gene therapy might have to go through multiple gene therapies. So often we see that often it will be required that a person goes through multiple gene therapies. Then again, we have the immune response sometimes, that for our immune system, the the world is very simple, that anything that is foreign, anything that's not a part of the body, kill it, destroy it. So everything that is not a part of the body is an enemy. So whether it was a good gene or a bad gene, the immune system doesn't care. Many a times, not always, but many a times, the immune system does not care. And if it just sees something new, it will try to destroy it. So that's why many a times when a new gene is being introduced into the body, perhaps the immune system itself will work against it, and it will destroy that new gene. So that's why many a times the immune response itself, it could be it actually could attack the new gene. It will might see it as an invader and simply attack it and destroy it. So that's why sometimes even the immune system can reduce the effectiveness of the gene therapy. Then sometimes what we see is that there are certain diseases which are multi-gene disorders, that the problem is not only with one gene, but rather there are multiple genes which have the problem. And because of that, again, it becomes very, very difficult for us to solve those kind of problems. For example, diseases, high blood pressure, Alzheimer's, arthritis, diabetes, they're all problems, they're all diseases that affect multiple genes. So multiple gene therapy is going to be extremely complex and almost impossible. So that also becomes one of the challenges many a times. So that's why there are all these three problems. Plus, right now, it's also very costly. Right now, the treatment is also very costly, and that also becomes one of the challenges with regards to gene therapy. So although we have seen limited success, we'll want more success, we'll want more stability out of it, but there are all these concerns that we need to address so that we can do better with regards to gene therapy. Then after that, there is another concept, which is the concept of three parents baby. Now this is a concept which is used in some unique ways or in certain unique situations, and that is when the mitochondria that belongs to the mother cells are unhealthy. So if the mitochondria in mother cells, not the father cells, the mother cells, if they are unhealthy, then in that case we try to use the method of three parents baby. Now what exactly is this method and why are we using this method? So first and foremost, understand this, that mitochondria that we have in our cells, and we discussed about mitochondria yesterday also. So mitochondria in our cells, they are always transferred from the mother to the child. Father's mitochondria are never transferred to the child. It's always the mother's mitochondria. So even if the father's mitochondria are diseased, doesn't matter. Why doesn't matter? Because it will remain restricted to the father. It will never be transferred to the child. But in case of mother's mitochondria will be transferred to the child. So if the mother has unhealthy mitochondria or diseased mitochondria, the same diseased mitochondria will also be transferred to the child. All right. So that is why this kind of a method will be helpful when mother's mitochondria are diseased. Now, what is the process? So the answer to why is, so our answer to when do we use it? We use it when the mother's mitochondria are diseased. Why we use it? Because mitochondria are always transferred from the mother to the child, never from the father. All right. So mitochondria are always transferred from the mother to the child, never from the father, and that's why if mother's mitochondria are diseased, then we'll have to use this method. Now, when we look at this method will be useful then. Now, how exactly do we use this method? We first take away the nucleus from the mother's cell, and we will take a donor egg cell. We'll take a donor egg cell, and again we'll remove this mitochondria from the donor cell. We will remove the mitochondria from the donor cell, just like what we do with cloning. So we'll remove the nucleus from the donor cell, and the nucleus from the mother cell, the original mother cell, will be transferred inside the donor cell. And then the fertilization will happen between the original mother cell and the original and the father cell, because the nucleus of mother and father are being retained, nucleus of the donor has been removed. So that's why the genetic properties, the specific properties that the child will have, will still be of the original mother and the father, because nucleus is of original mother and the father. So that's why even though there is a donor egg cell, the child will not have the properties of the donor. The child will have only the properties of the original mother and the original father. So that is why this is something that is worth noting, because there already has been a controversy that this was something that was uh, brought to the courts in the UK, where it was said that the donor should also have the claim of the child, because the egg cell is coming from the donor. But then this is where the courts had said that no, because the properties are only being inherited from the original mother and the original father, and donor could have been anyone. It could have been any healthy female cell or any healthy female egg cell. So that's why it doesn't matter, and that's why donor cannot have the claim over the child, only the original mother and the original father. So what are we doing? First, we take out the nucleus from the unhealthy mother cell or the mother cell with unhealthy mitochondria. So first the nucleus will be removed, then we take the donor cell, and from the donor cell we'll take away the nucleus, and the original mother's cell will be replaced inside the donor egg cell, and then the fertilization will happen between the donor egg cell with mother's nucleus and the father cell. So that is basically what has been shown in these three steps in this particular scenario. Now the question is that why nucleus? The problem was with the mitochondria. So why not replace the mitochondria? Why are we using an indirect method of uh, replacing the nucleus instead of mitochondria? Can you think of a reason? Yes, Ravi, you are correct. Nikki, you are correct. So basically, the thing is that mitochondria are multiple in number. There are multiple mitochondria and there is only one nucleus. So replacing one nucleus is easier as compared to replacing multiple mitochondria. So that is the reason why. That's the reason why we do not really replace mitochondria, but instead we replace nucleus. Also, it's something which is established procedure. In cloning, we already have been using it. Already certain similar process has been used. So that's why we can use that easily as compared to the complicated replacement of mitochondria. Right, so there are different names also that are given. For example, mitochondrial manipulation technology, or 3% in vitro fertilization, or another name that is used is pro-nuclear transfer. So this was also a question that was asked in your paper, pro-nuclear transfer. I'm sure you know the answer to this question, but let me still give you the poll here, just in case you have from the mother. So that's why the correct answer here will be, so pro nuclear transfer could have been very confusing at the time of the examination, I understand, because this is a very less used term. We use three parents baby or 3% in vitro fertilization or mitochondrial manipulation, etc. These are more used term, and pro nuclear transfer is a lesser used term, but they had given this in your paper. Now coming to GM mustard, a variant of mustard that has been often in the news in the last 3 4 years. We have seen it. So there is this variant of mustard, that hybrid variant of mustard that was developed in India, called as DMH11 or Dhara mustard hybrid 11. DMH11 or Dhara mustard hybrid 11. Now this DMH11 has a higher productivity, has a higher yield as compared to normal variants of mustard, and it has been under production, or I mean, we have been working on it for a very long time. 2011 and 2014 is where a lot of tests happened, and we saw that it is able to give almost up to 25 to 30% more yield as compared to best of the variants of mustard like Varuna currently that are grown in the country. And it is something that was developed in Delhi University Center for Genetic Manipulation of Crop Plants, which is located in Delhi University. It's situated in Delhi University. So that is where we had developed it, and we saw that the productivity is better. Productivity can be up to 25 to 30 times better. Sorry, 25 to 30% better as compared to all even the best variants of mustard that are being grown. So that is why in 2018 it was about to be uh, introduced into the Indian markets. The seeds of DMH11 was about to be made available for the Indian farmers. It was given a go ahead by the Genetic Engineering Appraisal Committee and also by the government of India. But then the Supreme Court intervened, and the Supreme Court had put a stay on the release of DMH11 seeds. Now, there are certain reasons that were cited that why this was done. Because first and foremost, until now, what we have seen is that most of the genetic modification that has been done in India, it has been done on cash crops, in on commercial crops more rather than on food crops. Now, when GM mustard is being grown, it will be for the first time, you can say, where at a very large scale, a food that is hugely consumed in India, a crop that is usually consumed in India, mustard, whether as seeds of mustard or mustard oil or in any form. So, for the first time, it is something which is so widely used in almost, I can say, almost every household in the country, right? So, that's why it was something that has to be taken into account, that it should not have any uh, the issues that were raised by Supreme Court. First, we have to check for long term health issues, that although some tests have been done, but we have to be very sure whether there are any chances of any long term impacts on human health. We Until now, whatever tests have been done and has been found to be safe, but can there be a problem 10 years later? Because there are issues in the past where we have seen this happen, that something that did not look to be dangerous in the short term was found to be dangerous when looked at it at 10 years or 15 years time period. So that's why that was one problem. And then the issue with biodiversity, the issue that it can create for biodiversity. Now, in what sense biodiversity issues are being discussed here? Now, when it comes to biodiversity related issues, basically the problem that was discussed is that right now India has almost 66 variants of mustard. All right, 66 variants of mustard are being grown in India. Once you introduce DMH11, obviously a lot of these mustard variants will not be grown anymore in the country. Correct? They'll not be grown anymore in the country, and instead, uh, everyone or every farmer will prefer DMH11. Obviously, it has a better productivity, so why not? So that, then what will it do? It will decrease the biodiversity of mustard, because many of the mustard variants will never be grown again. So that decreases biodiversity, and decrease in biodiversity is never good. Whether it is species diversity or genetic diversity or biological diversity, it is never good. So that's why these kind of concerns were raised, and that's why Supreme Court had put a stay, saying that look, uh, Europe already has put a stay on it, so why are we introducing it? The government side was that in the US already this very similar variant is being used, in other some of the Asian countries also already it is being used, so we can also safely introduce it. But the Supreme Court's stance was that Europe has banned it. So on one hand, you are saying that this country has not banned, but I also have an example that this country has banned it. So do more studies on it. So a committee was set up which would study DMH11 further and come out with its report. So last year they came out with the report saying that everything is good. There's no problem that we see with DMH11. So that is basically what we saw, and until now the government has not released it yet. It's still under consideration by the government, and they have not given any go ahead to DMH11 yet. So it is still under consideration as of now. This was a question, this was asked in your 2019 paper, a very simple question actually, that gives the plant the property of pest resistance to a wide variety of pests. Now, this kind of pest resistance from a soil bacterium is more property of the bt cotton. It is coming from bt cotton, so that's why this is not really about the hybrid variety of mustard that we are creating here, because there is no bacterial dna that is being transferred to this dmh11, so this is wrong. GM mustard has genes that allow the plant's cross pollination and hybridization. Now, this is correct. And this was also raised as a concern that because cross pollination and hybridization is possible here, so it may so happen that the same genes that you have transferred, or the genes that you have in this DMH11, might also get transferred to some of the other plants that are in the nearby areas. So, what if there is, say, any negative impact on the other crops? So that also was raised as a possibility. So that's correct. GM mustard has been developed jointly by IARI and Punjab Agricultural University. What we discussed is Delhi University. Now look at the options. You don't even need to read all these three, as some of you already have mentioned also. Statement number three is here, here, and here, and this was the easiest of the three statements in this particular question. So only by knowing about statement number three, you can get to the answer, and you could mark the answer. So statement number three made it easy. Otherwise, it could have been a difficult question. Otherwise, because the first two statements are a bit of technical statements and not very easy to mark as correct or wrong. So that's why statement number three actually makes it easy. So those were the days when a lot of questions could be answered with the help of elimination method. Right. So coming to the last topic of discussion in biotechnology, and this is dark biotechnology. Now, when we say dark biotechnology, the meaning of dark biotechnology is when you are misusing biotechnology, when it is being used for a negative purpose, then it is dark biotechnology. So whether it is bioterrorism or making of bioweapons or anything against doing something against the crops, negative impact on biodiversity, negative social impacts, anything, all these negative aspects of biotechnology will be considered as dark biotechnology. Now, in case of dark biotechnology, this is something where we are, that's why, talking about bioterrorism, biowarfare, biocrimes, anti-crop warfare, and so on. Now, in a sense, that's why the negative aspects of biological science and tools will be considered as a part of dark biotechnology. Now, in these cases, what we basically see is that there are a lot of ways in which biotechnology could be misused. For example, when we talk about bioterrorism or making bioweapons, so bioweapons is something that we partially discussed in DNA fingerprinting as well. But in case of bioterrorism, now how bioterrorism, what may happen is that, let's say, some kind of microorganism or some kind of pathogen might be unleashed, maybe on humans, that might cause death and destruction in human population, or even if it is done against even crops. For example, let's say a country like India is widely or hugely dependent on its agriculture. A lot of population of the country is dependent on it, for its livelihood is dependent on agriculture. So if, let's say, some kind of pathogen is released against crops in India, that also leads to wide destruction and will have a huge negative impact on the Indian economy and at an individual level to a lot of people. So even that will be counted as an act of terrorism. So that's why it all will be counted as bioterrorism. Similarly, they can be used to modify or enhance the performance to cause diseases and spread in the environment also. So we know that in all these cases, bioterrorism and bio warfare or making bioweapons is actually possible. Then even regeneration of extinct pathogens. You might have seen the news very recently, almost 5 6 months back. It was in the news that a very old virus, has been regenerated by the scientists. So it was not a pathogen, but it was a virus which was actually uh, it was actually uh, uh, regenerated by the scientists. So we know that it is possible that we can do that. Now, if you imagine how it can be used in a negative sense, So let's say if you consider something like smallpox, something that has been eradicated now, because there was wide vaccination that happened all across the world so that we could eliminate smallpox. But we still know about pox vaccine. There is chicken pox. There were cases of monkeypox a couple of years back. So we know pox virus, and we know what is the structure of the smallpox virus. What if in the lab tomorrow somebody develops it and unleashes it on population? Today's generation has not been vaccinated against smallpox. So that's why if let's say it becomes if let's say somebody actually does it, it can become an epidemic or even a pandemic for the world today. So that's why it's all possible and it all can be done, and it can be a very dangerous scenario. Then the effects that it can have on biodiversity. We discussed about the case of GM mustard, or how DMH11 can have a negative impact on biodiversity. Apart from that, what we see is that because of uh, biotechnology, our focus of biotechnology is only on a very few species, the species which are useful for us, right? Only certain crop varieties, only certain animal varieties, and that is where we are concerned. 99.99% of the species that exist in the world, we don't care about them because they are not so called useful for us, right? So that's why we are not even concerned about them. So what happens is that you are trying that these small number of species, they should be healthy enough, they are getting all the benefits of biotechnology, and they would survive. But a lot of others, or almost all the other species, will not, because their biodiversity will keep on decreasing and nobody will care to save them. So that's why, because of our focus only on a few species, we see that it can lead to decrease in biodiversity also. So that's why that's all a problem that actually can happen. Then there is the case of terminator genes. Now terminator genes are special genes which are introduced in the crops, which are introduced in the seeds, so that seeds germinate only once. All right. They'll so terminator genes, these are genes artificially introduced in the seeds so that the seeds germinate only once. All right. The seeds germinate only once. So that's why we say that these are called as terminator genes. Now these terminator genes are very dangerous, because what happens is, let's say as a farmer, you go to you go to the market and you buy some seeds, and you use those seeds for your agricultural field for that particular season. And let's say you see a very good growth, right? Quality wise, quantity wise, you see a very good growth. So obviously you'll want to preserve those seeds for the next season as well. So you do that. Let's say you do that. But in the next season, when you sow the same seeds, you see that the seeds don't germinate. In the last season, you saw such a good growth, and this time you see that the seeds are not even germinating. Why? Because the company from where you had bought those seeds, they had used terminator genes. They did not obviously tell you, but they used terminator genes, so that the farmer has to come back to the company again and again to buy these seeds. And that's a big problem, because then what happens is that the next year again you'll have to go back to the market, you'll again have to buy those seeds, and this time the prices of the seeds would have increased because they have already made you dependent on them. So that's an unethical practice. But many of the big seed companies, agricultural companies, have actually used this practice of using or introducing terminator genes in a manner that the seeds only germinate once. So that becomes a big challenge, because it may lead to a lot of wealth drain from the farmers, because they'll have to keep buying the seeds over and over again instead of reusing the same seeds from the older crop. So that is one problem. At the same time, what may also happen is that these terminator genes uh, they may cross pollinate, and may react. All right, they may cross pollinate and may react with some of the other plants as well, which are growing in the same field, because many a times the farmers do not just grow the same crop, but rather there might be multiple crops which are growing together. So these terminator genes may also transfer to some of the other crops which were growing in this particular area. So that also increases the chances of the same terminator gene also transferring to other crops, and those other crops also getting affected. So not only the crop which had terminator gene gets affected, but even the others get affected. So that becomes a big challenge. So overall, that's why we need to understand all these issues with regards to terminator genes and how exactly they can also cause problems, right? So this is the end of this particular chapter. Now, before going on to the next chapter, one thing that has been pending is that we have not discussed a lot of mains questions. So either we can do it today, or because from space technology and energy, I've brought the questions, or let's do it today, right? Let's do it today, that these are the questions that have been asked. I have not included the 2023 questions here. We'll separately discuss the 2023 prelims and mains paper towards the end. After all the chapters are done, the 2023 prelims and mains questions we'll discuss towards the end. All right, so now, looking at space technology, as I told you, that space technology is a chapter from where every year one question comes. So one question did come in 2023 also, and you'll see that same has been happening every year. Just that two COVID years is when we did not see questions from space technology. 2020 and 2021, they did not ask. Otherwise, if you see 2022, 2019, 2018, 2017, every year 2016, 2015, every year they are asking one question, right? Every year they are asking at least one question from space technology. So that's why I was saying that this is the most important chapter of everything that we are discussing now. If you see 2022 question, yeah, this was about the James Webb Space Telescope, and a topic which was probably the most important topic and the most widely discussed topic of 2022. We expected that a question would come in the prelims examination. They did not ask any question in prelims, so they asked a question in mains. Now, if you look at the question, and this is where reading the question becomes very crucial. Reading the questions carefully becomes very crucial. That if you see, what it says is, launched on 25th December 2021, James Webb Space Telescope has been much in the news since then. What are its unique features which make it superior to its predecessor telescopes? What are the key goals of this mission? What are the potential benefits that it holds for the human race? So if you see, there are three questions, right? There are three questions that you have here. And very often it's easy for us to neglect one of the questions. We might, so what may so happen is that you know about James Webb and you just start writing everything that you know about James Webb Space Telescope. So that can never be your approach. That should never be your approach. Always look at the specific questions or the specific parts of the question that we have, and how exactly do you have to answer them. What are the points that you are going to use there? So for example, first it talks about the unique features that makes it superior to its predecessor space telescopes. Now, when it has mentioned predecessor space telescopes, the mention of those terms or those names is very important. For example, Hubble Space Telescope is seen as the predecessor of the James Webb Space Telescope, or even up to an extent, the Spitzer Space Telescope is also seen as a predecessor of this telescope, right? So now what happens is that if you are mentioning these names, James Webb and Hubble, so then it, in that sense, to the examiner also it shows that you are prepared, that you know your facts right, you know your data right, and that also gives you a positive impact. Obviously it will have a positive impact even on the number that you're getting, the marks that you are obtaining in that question. So that's why the unique features, what are the most important unique features? The first and the foremost, nothing better than this, is about the use of infrared. This is an infrared telescope. Now, because it's an infrared telescope, it can actually see better. All right, it can actually see better and can see farther. It can also see farther. How exactly, all that we'll discuss when we go to IT and communication chapter. There we'll discuss about infrared rays, and it will make it clear for you that how exactly it can see farther. Secondly, it also has been placed on L2. Hubble Space Telescope was placed at uh, the low Earth orbit. So because of that, there will be times when it is towards the sun, then it will be away from the sun. So that's not good. When it comes to, let's say, using of that orbit for space exploration. But in case of James Webb Space Telescope, because it has been kept at L2, it is always shielded from the sun, and hence it will be able to give you better results. So that's why that's there. And then apart from that, there are other uh, features. For example, uh, the sensors that have been used, the shields that have been used, all these are far more superior as compared to anything that we have seen until now. Then the goals of the mission. The goals of the mission basically are more about better understanding of the universe, better understanding of the universe, and the origins of the universe. Most importantly, also the origins of the universe. So this will give us a lot of this will basically give us a lot of uh, huge advantages, and that is what the third part of the question wants us to answer. What are the potential benefits that it holds for the human race? Now, when it comes to the potential benefits, basically what happens here is that there are a lot of things that we might know better about the universe. We might know better about the origins of even the earth and the other planets in the solar system. We might know about the unknowns of the universe that we have never seen. We might come across something new that was never observed, and things that we already start seeing, right? We have already started seeing that some of the stars that they have observed, those stars, according to whatever we know until now, should not have existed at all, but we have seen those stars. We have taken pictures of those stars with the help of this telescope. So that's why a lot of unknowns of the universe, for example, even let's say dark matter. So maybe it will give us some clue about what is dark matter and what is dark energy. So that's why there are all these things that can be taken as a potential benefit of all these things. So there are all these points that you have to write, but before this, obviously, you'll give an introduction here. An introduction will be simply stating the facts about it, that James Webb Space Telescope is an infrared telescope that is seen as a successor of the Hubble Space Telescope, one of the most important launches that have happened in the past few years. And there are a lot of, basically, you can summarize everything that's there in this question. So that will basically become an introduction for you, and then start writing everything uh, in maybe a point wise manner, or in a uh, paragraph if you want. So you can do that. Now, the only thing that matters in writing answers to all these questions is that have you addressed every part of the question or not. And this is where we see that a lot of students suffer, because you'll start writing because you have some information in mind, and you just start writing your answer based on that. So that's why focus always on all these kind of smaller things also. Then the next question, if you see, what is India's plan to have its own space station, and how will it benefit our space program? So this is something that was in the news in 2018, because in 2018 there were some news flying around that India might join the International Space Station. But then India had said that we are working on our own plan for having our own space station, and we are trying to target 2030, that by 2030 India will have its own space station. And what was said is that right now we are working on the Gaganyaan mission plan. And once the Gaganyaan mission is successful, after that we'll start working on our own space station. So this is what was said. when it came to that, and that's why it was also, you can say in a manner, current affairs based question. So now when it comes to this kind of question, the when, because it is again talking about the benefit, that what is the benefit. So what is the plan? We discussed that this is the plan, that by 2030 we want to have our own space station, and right now we are working on some of the other key important uh, missions like the Gaganyaan mission, and after the Gaganyaan mission it was announced that we'll start working on our own space station. Now what are the potential benefits? What are the potential benefits that it will have for our space program? So first and foremost, we'll be able to do better research. Staying in the low Earth orbit, we'll be able to understand the space environment better. So it will give us a better understanding of the space environment. At the same time, a lot of microgravity experiments can be conducted. That in those kind of situations where the gravitational pull is very weak, how does the space react? Even biologically, how do plants and animals, or including humans, react? How does the human body react to such a situation? So that will give us a better insight of everything, how it works, and the basic uh, relation between the physics and the biology of the body. So there are all these things, plus it can also act as a support system for all our satellites, especially the satellites that are placed in the low Earth orbit. So there are multiple benefits that we can draw out of all these kind of technologies with our space station. Then after that, there is this question, a very simple direct question based on IRNSS. Why is IRNSS needed? How does it help in navigation? So why is it needed? We have a historical context to it. That first and foremost, we don't want to be dependent on GPS only, because GPS at the end of the day belongs to US. So we don't want to be dependent on it. We want our own technology for security purposes. Also, it is very necessary to be dependent on on our own technology, plus our learnings from the past, whether it is bit to 1998 or whether it is 1999, 2 years which made us realize the importance of having our own navigation system. That in 1998, when we were doing the nuclear test, we were being spied upon by, with the help of GPS satellites. The US was spying up on us, because it had some news that something of this sort is about to happen in India. But we were able to somehow mask ourselves from it, and we were able to carry the nuclear test successfully. If at all the world knew, if at all countries like US knew, they would have put so much pressure on India that those kind of projects would have never succeeded. So that's why it was very important for us to keep it a secret, keep it a top secret that nobody knows of, and thankfully we were able to do that. But we were being spied upon with the help of GPS satellites by the US. 1999, this was the Kargil War. Now, during the Kargil War, India asked the US for some GPS information of the Kargil region, because you might know about the war, that how we were at a disadvantage because we had to climb up, and the Pakistanis were already sitting at the top. So it made it much easier for them uh, to fight against us. So we wanted the GPS information, the locational information, so that we could strategize better. The US refused. The US back then refused, said that will not provide the information. So these 2 years back to back, these two incidents back to back, made us realize that we can't really rely on them, and we'll have to have our own GPS, or other navigation system. 2001, the project basically started. 2003 to 2013, we developed all these satellites, and 2013 onwards, IRNSS satellites started getting launched. How does it help in navigation? We discussed about the role of the geostationary satellites and the geosynchronous satellites, and how you use the method of triangulation. So we discussed all these things, so you basically can write all these things in writing an answer here. Then after that, so basically you can say that when it comes to why it is needed for our internal security, for covering the Indian Ocean region, as I told you, that up to 1500 kilometers beyond the Indian borders, right? Plus it will also help some of our neighboring countries. Also it will help us have better relations and strategic relations with the countries. So in all these cases, it will be very, very useful. Next question. India has remarkable successes in unmanned space missions, including Chandrayaan and Mars orbital missions, but has not ventured into manned space missions, both in terms of technology and logistics. Examine critically. So this was the time when we were talking about the Gaganyaan mission for the first time. That now we'll be working on a human space flight, which has never happened in India until now. So that's where the challenges, so technological challenges and the logistical challenges. So it was also pretty much a current affairs based question because it was in the news back then. So security wise, we know the issues that we don't want any loss of life, so we'll have to take utmost precaution in ensuring that nothing happens to anyone who's involved with this process. Similarly, at the same time, how would you design the suit, how would you design the helmets, how would you design the launch vehicle and the space vehicle, the crew module, the testing of the crew module, and all these kind of challenges, plus the cost, the logistical challenges as well. The cost is also going to be huge. For example, in Gaganyaan mission, we know that the total budget is approximately 10,000 crores. So that's why it's a huge mission. It's a very costly mission. So that's why there are all these challenges that we have with regards to this. Then after that, the next question, if you see, a pretty simple, straightforward question. Discuss India's achievements in the fields of science and technology, and how the application of this technology has helped India in its socio-economic development. How it has helped in socio-economic development. So when it comes to achievements, you can simply start mentioning all these achievements historically, right? Basically, the timeline of what happened when. So we can talk about that, or the other way could be you keep talking about all the different things that we have done, how the earth observation satellites or the remote sensing satellites have helped, how the navigation satellites with IRNSS has helped, how all the communication satellites in establishing mobile phone communication or radio television communication and all that has helped, or telemedicine or teleeducation etc has also gotten its help. So you can mention all these things, right? You can mention all these things, plus the socio-economic developments. So socio-economic development, we know that it has helped our agriculture, it has helped in establishing better communication, it has helped in developing a lot or generating a lot of new employment generation, it has helped the entire space market developing in India, it has generated a lot of revenue, a lot of startups have developed because of that. So a lot of things that we have already discussed, and you can keep writing all these things, but remember that it has to be written in 200 words. That is one of the challenges you might have in writing such an answer, where even if it was about writing 500 words, you could have still written the answer, right? Then after that, what do you understand by standard positioning system and precision positioning system in the GPS era? Discuss the advantages India perceives from its ambitious IRNSS program. So again, something that we have discussed. So you just have to write all of those things. You have just all that information. You simply have to discuss here. If you remember, we discussed that there is standard positioning system and the restricted service. So that restricted service is also called as the precision positioning system. So you simply have to write that and why we need it. And then after that, IRNSS, again, similar question to what we just discussed. Second, energy. Also a topic from where a lot of questions come. Of late, at least, we have seen that the number of questions have risen from this topic. Now look at the first question. Do you think India will meet 50% of its energy needs from renewable energy by 2030? Justify your answer. How will the shift of subsidies from fossil fuels to renewables help achieve the above objective? Explain. Now, the first part, do you think? The answer is not yes or no. Of course, it is not no. You cannot say no. India can't. You cannot say no. Obviously, you cannot say no. But the answer is not even yes. You cannot simply say that yes, India will, and think that okay, government will be very happy with you and give 15 out of 15. No, that's not the case. When it comes to these kind of questions, that do you think something like this will happen, you have to be objective about giving these answers. That you will first have to talk about what's the present scenario, that this is where we are, what's the future, this is what we are doing, and if we do everything as required and if we do as per the plan, then obviously we'll be able to reach the required targets. So this is the tone that you should take in writing such an answer. Instead of simply writing yes or no, say that this is where we are, these are the programs, policies and schemes that the government has, and these are the targets. And if we go in this direction, if we whatever we are doing, if we continue on this path, of course we'll be able to achieve the targets. So you have to take this kind of a tone in writing such an answer. So when it comes to justifying the answer, it will be about everything, every scheme that has been launched, every target, and how are we making meeting the target, taking the specific names. What is the national solar mission, or the Srishti scheme, or how the mega solar power plants are going to work, how the floating solar power plants are going to work, what are we doing in wind energy, what are we doing with offshore wind, what are we doing with hydrogen, what are we doing with green hydrogen. So there are so many things, right? So if you are able to mention all these specific names, it will show your preparedness, and that is how you get a better answer and a better marks. Will the shift of subsidies from fossil fuels to renewables help? Of course it will, because whenever you are subsidizing something, you are providing a financial help. So whatever sector you provide the financial help, the sector will grow, because people will come there, people know that the government is providing that financial help. So of course it will help, right? Then if you see to the next question, it is about the Nobel Prize in the invention of blue LEDs, and how has this invention impacted the everyday life of human beings. Now blue LED is everywhere. Your mobile phone or laptop screen on which you are watching this lecture is blue LED. LEDs that you have around your houses, based on blue LEDs. Medical applications, blue LEDs. So everywhere we see that blue LEDs basically are available now. And we know, and here this is where it becomes very, very important, that the schemes that we have in India, for example, the Ujala scheme, that how Ujala scheme has been very, very important, how the government also coming forward and just distributing free LEDs in the rural areas, so that LEDs will be everywhere, and bringing the cost down of the LEDs. So all these kind of things have to be mentioned, plus the longevity of the LEDs compared to the incandescent bulbs, that has to be mentioned. The amount of power saving that you do, because they consume a lot less power as compared to the incandescent bulbs. So there are all these things that you'll have to mention when it comes to this kind of a question. Then with growing, if you look at these two questions, both of them are based on nuclear energy. Both of them are based on nuclear energy. So, and are very similar. With growing needs, should India keep on expanding its nuclear energy program? The answer, of course, is yes. We cannot say that India's energy needs are growing, but we should stop using nuclear energy. But the second part talks about the facts and fears, right? That what are the advantages and what are the problems, what are the fears that we have. So you'll have to write about those fears, that chances of nuclear meltdown, accidents, leakage, etc., the disposal of nuclear waste. You'll have to talk about all these things, but end on a negative sorry, never end on a negative note, end on a positive note, and talk about what can be done potentially so that these issues do not happen at all. How can we ensure that these issues do not happen at all? So you'll have to write about all those things here. Plus also briefly try to mention fusion. Briefly try to mention fusion as well, because that's the future. So that's why in future this is also a possibility, you should write about that. So fusion, thorium, all these terms should definitely come in writing this answer. Then give an account of growth and development of nuclear science and technology in India. What is the advantage of the fast breeder reactor program? So we discussed about fast breeder reactor and the advantage that it has. And here you have to simply write about India's nuclear program. That what India's nuclear program is and how we want to use India's nuclear program for getting better energy and more energy out of nuclear technology. So that's why these are all these kind of questions. So all these questions are simple. I just want you to write. I just want you to write answers to these questions, because me discussing it, I'm telling you, I can give you all the points, still does not matter, because ultimately you'll have to write answers to newer questions in your own words. So you have to develop a habit to do that. So please ensure that you get into a habit. I mean, even if you can just write one answer a week, even that is good if you just start. But unfortunately, what we see is that despite whatever we say, students are never bothered about it. And you think that, okay, fine, I've read enough, I've attended all these lectures, I've gone through the notes, and that should be enough. It will never be enough unless you have started writing on your own. Unless you start writing on your own, whatever strategies we discuss, whatever points we discuss, it never matters. It only and only matters when you take that step and you pick up the pen and you start writing your answers. So please make it a habit. Please ensure that you start doing that, because otherwise you will never be able to crack the mains examination, and you will realize after you commit that mistake. So please don't do that, and start writing your own answers. So let's not start the new topic. Let's start the new topic tomorrow. Anyway, we have the webinar now at 8:30, and I only have to take that webinar. So I also need a break of like 10 minutes, so that again I can come back after the break and I can take the webinar also. So today we have the science and technology and environment webinar that I'm only taking it. So see you there. See you again. The same face you'll have to manage for 2 more hours. So that will start by 8:30, maybe between 8:30 8:35. I'm showing you the leaderboard as you have requested.